you for joining this industry update webinar. My name is Scott Baver, and I'm a Senior Director on the Medical Affairs team here at Apellis Pharmaceuticals. Today, it's my great pleasure to introduce to you Dr. Rup Tandon, who will be speaking on the role of complement and complement component C3 in ALS, as well as discussing the MERIDIAN trial of Pegcetacoplan in ALS. But before I introduce Dr. Tandon, I'd like to give a brief introduction to Apellis for those of you who are not familiar with the company. So Apellis is a biotechnology company from the Boston, Massachusetts area. And at Apellis, we're committed to developing transformative therapies for people living with a broad range of debilitating diseases by controlling a part of the body's immune system called the complement system. So Dr. Tandon will describe the complement cascade in more detail today, but our work is really rooted centrally at the level of C3 in the complement cascade. And we at Apellis, we believe that targeting C3 is critical to reducing uncontrolled complement activation at all aspects of disease. And that targeting C3 has broad platform potential to treat many diseases where patients have few or no treatments options available. And with our lead compound, Pegcetacoplan, which has been recently approved in a rare hematological condition called PNH, we're really leveraging the science of C3 inhibition to advance treatments in several diseases with significant unmet needs across the areas of hematology, ophthalmology, nephrology, and neurology in diseases, including ALS. So with our courageous science, creativity, and compassion, we really are committed to delivering therapies for people with complement-driven diseases. Okay, and now I am very grateful to introduce Dr. Tandon. Dr. Tandon is an attending neurologist at the University of Vermont Medical Center in Burlington, Vermont. He received his medical degree from King George's University in Lucknow, India. He did his postgraduate training at King George's Medical University and then to the Royal College of Physicians and University of Birmingham in the UK, and completed his residency in neurology at the University of Michigan and did a neuromuscular fellowship at the University of Vermont. So currently, Dr. Tandon is a fellow at the Royal College of Physicians in the American Academy of Neurology, and his clinical and research interests include neuromuscular diseases, and in particular, ALS and myasthenia gravis. He served as a principal investigator on numerous clinical trials. He's an investigator on the MERIDIAN study. He's chaired scientific sessions at national and international venues and is an ad hoc reviewer on numerous journals. So also of note that Dr. Tandon has authored and co-authored more than 325 abstracts, reviews, peer-reviewed manuscripts, and book chapters on a range of neuromuscular diseases, including ALS and myasthenia gravis. Okay, now I'm going to turn it over to you, Dr. Tandon. Thank you very much.
Thank you, Scott. And first of all, I'd like to thank Apellis Pharmaceuticals for giving me this opportunity, and also to MDA for organizing this webinar. So the role of complement in ALS and the MERIDIAN study of Pegcetacoplan and sporadic ALS is what the topic is today. So I will take this topic into 4 different parts or sections. In the first part, I will discuss the complement in general. Second part, I will discuss the evidence for the use of, for the abnormalities of complement in patients with ALS and in murine models of ALS. Thirdly, I will talk briefly, very briefly, about Pegcetacoplan, and then lastly, I'll introduce the MERIDIAN study of Pegcetacoplan in patients with ALS. Next slide. So this event, as I mentioned, is sponsored by Apellis Pharmaceuticals. It does not provide CME credits for physicians, and Pegcetacoplan has not been approved for ALS, but as Scott mentioned, has been approved for the treatment of paroxysmal nocturnal hemoglobinuria by the FDA. Next slide. So I think I've done this. Next slide, please. Next slide. So the complement system is a very complex network, and it serves immune functions. It is a network of proteins, many proteins, the key of which is C3. and a network of membrane-expressed receptors and regulators, which are present in the plasma, in the tissues, and in this instance, in the tissues of murine models of ALS and in human ALS patients, and also within cells. The complement system regulates and coordinates the activity of the innate immune system and the adaptive immune system. And this is to provide integrated host defense response mechanisms. So the innate system is the system that is activated, that we are born with, and is activated in the instance of an acute infection or acute cell loss or degeneration. And it is, we will talk about that in a few minutes. It's composed of multiple cell types, including complement, which is one of its major constituents. The adaptive immune system is a system that we create as we go along and we are exposed to infections or other noxious agents in the atmosphere or in the environment. And that consists of mainly the T cell and B cell system, but then there are many others, including an overlap between the immune system and the adaptive system, such as the NK cell or natural killer cell and macrophages, which are phagocytic cells predominantly. So even though the complement system eliminates pathogens without injuring the host body, it can cause, it's associated with numerous diseases, especially when there's uncontrolled activation of the complement system, some of which I think Scott mentioned, for example, paroxysmal nocturnal hemoglobinuria that I mentioned briefly earlier on, age-related macular degeneration that is also associated with complement activation, and then some other conditions or kidneys, etc., that are associated with abnormal complement activation. Next slide. So the complement cascade is what is spoken of when one is talking about the complement system. The complement cascade consists of basically three pathways: the classical pathway, the lectin pathway, and the alternative pathway of the complement system. And all these pathways, when they are activated as a result of injury or infection in particular, they lead to the formation of C3 and ultimately C5b9, which is the membrane attack complex that we all probably have heard about and know about as being a cellular destructive system that causes cell lysis and also inflammation and oxidation. So we'll talk about these systems. So the classical pathway is activated when antibody binds to antigen, and the C1q component of complement then binds to the Fc receptor of the antibody. And in the presence of C1r and C1s, it cleaves C4, another complement moiety, and C2 into C4ab and C2ab. Then C4b and C2a combine to form a protease called the C4b/C2a protease, which then cleaves C3. And then let's talk about the lectin pathway. So the lectin pathway is recognized by lectins, which mannose binding leptin is one of them, and collecting is another, and phycolins are a third kind of leptins. When they encounter carbohydrate or other toxic or infectious agents, they are activated to produce what are called mannose binding lectin associated serum proteases. And these proteases function as the protease emanating from the classical pathway to ultimately cleave C3. The third pathway is the alternative pathway, which is activated due to a spontaneous hydrolysis of C3 complement. And the C3 complement then binds to factor B to produce C3b, which then can act as a protease and cleave C3 as well. So what you see is that all three pathways ultimately converge on C3. Then C3 is actually converted to C5 through the activation of a protease, and then this protease actually cleaves C5 into C5a and C5b. Prior to this, C3 is also cleaved into C3a and C3b, C3A is involved in inflammation by binding to its receptor, and C3B is involved in opsonization, by which the invading organism, whether it be a bacteria or fungus or virus, is coated and then disposed of by the macrophage system in the body. C5b, or the membrane attack complex, is deposited on the membranes of the infectious agent or on cells, and it produces inflammation, lysis, and opsonization, thereby eliminating the infectious agent and sometimes producing disease in individuals, in humans in particular, like in myasthenia gravis. Next slide. So, as I mentioned, so in the three pathways, the central player in these three pathways is C3. And when C3 is activated, it causes the cleavage of C5 into C5b9, which is membrane attack complex. Now, you don't want C3 to be activated in the host system and cause host system damage. So there are many checks and balances of producing inhibitors of wayward activation of C3. And that happens in all these three pathways. So for example, in the classical pathway, the C1 inhibitor prevents excessive combination of C1q with antibody to form the protease that would then cleave C3. In the alternative pathway, there are agents that prevent the formation of MASPs, the serine proteases that can cleave C3. And in the alternative pathway, there are inhibitors that prevent the excessive hydrolysis of C3 and therefore ultimately excessive cleavage of C3. So these inhibitors are sort of check mechanisms to prevent the damage of the host cell or host cell and organs due to C3 activation. Next slide. Complement, next slide. Yes. So, so the key players, therefore, are the convertases, which convert C1 into C4 and C2 into C4A and B and C2A and B, producing the C4B, C2A convertase that cleaves C3. The other convertases are the C3B convertase that then acts on C5 to cause the C5 to be cleaved into C5A and C5B. And C5A is an pro-inflammatory protein where the C5B combines with C6, C7, C8, and ultimately C9 to produce the membrane attack complex, which causes inflammation, lysis, and opsonization. Next slide. And as I mentioned earlier, that to prevent host damage due to uncontrolled activation of these C3 and C5 moieties, there are regulators, which are the C1 inhibitor for in the classical pathway, and the CD55 factor H and CR1 in the alternative pathway, and then the factor I, factor H, and CR1, which prevent the excessive activation of C3 and amplification through the C3b to produce excessive cleavage of C3. Next slide. Next slide. So what about the role of complement in ALS? Next slide. So there's ample evidence that the complement system goes awry in ALS, both in the murine models of ALS and also in human ALS. And the complement system is involved with of course, the immune system, but also is closely integrated with the inflammatory system or the neuroinflammation, which are implicated in the pathogenesis of ALS. This includes not only the onset of the disease in ALS, where the immune system and the inflammatory systems are implicated, but also in the progression of the degeneration and the loss of function in the murine models of ALS and in human ALS. So in ALS, there are multiple mechanisms that have been incriminated in the damage to the motor neurons and death of motor neurons in the human condition. But one of the early inciting events is neuroinflammation. The neuroinflammation results from microglial activation, and the microglia produce a number of cytokines and inflammatory markers that produce inflammation, as also astrocytic proliferation with the formation of reactive astrocytes. So both the astrocytic proliferation and macroglial activation lead to significant neuroinflammation, including with the presence of the usual pro-inflammatory markers like IL-1, IL-6, TNF-alpha, etc., in the brains and spinal cords of murine models of ALS and in human ALS. The astrocytic proliferation also induces a cytotoxicity, which, as we all know, has been incriminated in the loss of motor neurons in ALS as well. So all in all, there's a toxic glial macroenvironment in ALS models of the disease and also in human ALS. What is less well known and less well studied until the last maybe 10 years or so is that the immune system also influences glia, especially the Schwann cells, the terminal Schwann cells at the neuromuscular junction and synapse. And that is the basis, I think, for what we are speaking about today in terms of reviewing the neuromuscular junction synapse maintenance due to in murine models of ALS and also in human ALS. So the components of the immune system and the complement system may be implicated in ALS. And I'm now going to review with you the evidence that the literature has as to why the complement system may be relevant in the etiology and some of the early changes that happen in ALS. Next slide. Okay, so most of this work is from murine models, particularly the G93A model of transgenic model of ALS, which is from the SOD1 gene. But more recent work has also used the TDP43 model and shown somewhat similar results as the G93A model of the murine ALS. So there's evidence suggesting that complement is involved in ALS in these murine models. So there's elevation of C3 in the serum, in the nervous system, the spinal cords, and in the CSF in ALS models. The progression of disease in these models is correlated strongly with C3 and the levels of C3. And that overactivation of C3, i.e. deposition of C3, occurs on Schwann cells and neuromuscular junction quite early in the course of the disease and murine models. In fact, even before there is any loss of axons in the roots, or even before there's motor neuron loss in the spinal cord. So this is a very early event in ALS, the deposition of C3 at the neuromuscular junction. And this C3 deposition at the neuromuscular junction with motor axon retraction in ALS is evident, as I mentioned before, the symptoms begin. And about the time that there's the innervation of the motor end plates in these animal models. Next slide. What about in human ALS? Well, in human ALS, there's upregulation of some of the proteins that we talked about earlier. There are complement proteins in the blood, in the plasma, and serum of patients with ALS. So on the left here is a series of two figures. The one on the left here is looking at C5a in the plasma of ALS patients and comparing that to healthy controls. And on the right, we are looking at C5b9, soluble C5b9 in the plasma of ALS patients as compared to healthy controls. And one can see that there is, even though there is some overlap, but there is an overall mean increase in C5a and in C5b9 in patients with ALS as compared to healthy controls. So there's evidence of activation and increase in levels of complement proteins in the plasma and also in the serum in patients with ALS. The other thing is that since C3 is the key central component in this model from all the three pathways, there's an increase in the levels of some C3 breakdown products like C3C1, C3C2A, and C3DG, which are breakdown products of complement proteins, especially C3, suggesting that there's increased activation of the C3 pathway in patients with ALS. So this was done in quite a large number of patients with ALS, and this was compared to the levels in normal controls, and the y-axis represents the degree of increase as compared to normal healthy controls in these various proteins in ALS patients. Next slide. What about CSF in patients with ALS? So this figure looks at the levels of C3 in the CSF of ALS patients and healthy controls, and is showing that there's an increase in the level of C3 in the CSF in patients with ALS as compared to healthy controls. Again, so in suggesting that increased levels of C3 proteins and the terminal complement protein suggest that complement-mediated inflammatory processes in ALS are at play and may be relevant in being able to be modulated by appropriate therapies. Next slide. So here, this cartoon is showing the deposition of C3 in the motor nerve terminal in ALS patients, but not in the control spinal cord and the control nerve terminal, and the deposition of C3 in the Schwann cells, the neuromuscular junction in patients with ALS, but not in control situations. So there is significant C3 and C3b, which is the activated product of C3 in the neuromuscular junction of patients with ALS. Next slide. So this is looking at the mouse model of ALS, the TDP-43 model of the mouse model of ALS, which has been more recently studied as compared to the D93A model. And this is looking at the accumulation of C3 in the spinal cords of these animals at various ages. So at 3 months, at 10 months, and at 16 months. So at 3 months, and comparing it to non-transgenic control mice and TDP-43 wild type mice. So at 3 months, there is a slight increase in the level of C3 in the spinal cord, but does not reach significance. But at 10 months, so these animals, the TDP-43 transgenic animals, have a much more protracted course of disease as compared to the D93A models. So here they develop disease by about 10 months or so, at which time there's an increase in the level of C3 in the spinal cord, which is significantly higher than that in transgenic animals, non-transgenic animals, and wild-type animals. And this increases even further when the animals reach an age of 16 months or so. So there's a 1.4 to 1.7-fold increase in C3 accumulation in the spinal cord in these TDP-43 transgenic animals. And in these animals, if you compare the grip strength, the force of the force of the grip strength in these animals, it is related to the levels of C3 in the spinal cord. So with higher levels of spinal cord, with lower levels of C3, significant reduction of the grip strength in the animal. So there's a direct correlation between C3 spinal cord levels and the grip strength in animals. Next slide. Similarly, in patients with ALS, when you look at the ALS functional rating scale revised score, ALS-FRS-R score, there is a negative correlation between C3 levels and C3b levels and the ALS-FRS score, so that in patients who had higher levels of the C3, they had higher ALS-FRS scores, and those who had lower levels had lower scores. Similarly with a C3b, which is a byproduct or end product of C3 itself. So complement protein levels are found to be significantly associated with ALS-FRS scores in human ALS as well. Next slide. Okay, so in summary here, complement C3 and its effectors are elevated in the serum and in the spinal cords and CSF of patients with ALS as compared to non-affected controls. Complement C3 and C3b localize it to the neuromuscular junction, and even before there is any clinical sign of disease in animal models, or there's loss of ventral root axons or motor neurons in the spinal cord, so it's an early event. The disease severity, as measured by the ALS-FRS-R score, correlates with elevated levels of C3 and C3 breakdown products, in this instance C3b, and that inhibition of complement pathways and complement should be effective in the treatment of ALS. Next slide. Next slide. So let's now come to the third part of the talk, which is the pegcetacoplan. So pegcetacoplan is an investigational polypeptide, synthetic peptide, which is attached to two ends of polyethylene glycol, or PEG40. And this product, pegylated cetacoplan, has a molecular weight of 43.5 kilodaltons. And the idea of coupling the pegcetacoplan with polyethylene glycol is that the PEG actually increases the solubility of cetacoplan and also increases the time it stays in the body. So there's a more longer effect, more therapeutic effect in the body. Next. So what does pegcetacoplan do? It actually is a significant inhibitor of C3. So we talked about the value of C3 in the genesis of murine disease and human disease in ALS. So what pegcetacoplan does is that it binds to C3 and inhibits it. Therefore, all the downstream events from C3 activation are also abrogated. So that you would expect that there is less formation of C3b, which is the C5 convertase, leading to the formation of MAC, or membrane attack complex. And there's also inhibition of C3a, which causes inflammation, and C3b that produces oxidation. So with C3 inhibition, there's a decrease in the production of a number of downstream products, including MAC, that lead to neuronal degeneration in ALS. Next slide. So what about the safety of this product? So there was a recent publication in New England Journal of Medicine that looked at the study of pegcetacoplan and eculizumab in patients with PNH. And this is a summary of the adverse effect profile of pegcetacoplan and eculizumab from that study. That shows that the number of adverse events, any treatment emergent adverse events, were the same in these two groups of patients. Eculizumab, by the way, is a C5 inhibitor, and APL2 or pegcetacoplan is a C3 inhibitor. Serious treatment emergent adverse events were similar between these two medications. Discontinuations were almost similar, just slightly more in pegcetacoplan group than in the eculizumab group. What was noticed is that there was slightly increased diarrhea in patients who got pegcetacoplan as compared to patients who received eculizumab, but these were mild events, and in the majority of events, this was a single event. A patient, there was a single event, so it was not clinically significantly relevant and led to no discontinuation from the study. So this is simply giving us an idea about the adverse effect or safety profile of pegcetacoplan as compared to eculizumab, which is the drug that has been used and approved in patients with PNH, and comparing the adverse effect profile of these two medications. Next slide. So the fourth part of the presentation is the MERIDIAN study in patients with ALS. So this is a phase 2 randomized double-blind placebo-controlled multi-center study to evaluate the efficacy and safety of pegcetacoplan in patients with ALS. Overall, it is a 116-week study, which is split into 6 weeks of a screening period, in which 228 patients will be evaluated, and then randomized 2 to 1. So 2 to 1, 2 patients receiving pegcetacoplan and 1 patient receiving placebo in a block design. And then this is the double-blind phase of the study, which will continue for 52 weeks. So patients who are eligible in the screening visit and the baseline visit will then be randomized 2 to 1 to receive pegcetacoplan or placebo. And then there will be an open label study for 52 weeks in patients who agree to participate in that open label study. Then as they come off treatment, there'll be a 6-week post-treatment follow-up in these patients. So there'll be 104 weeks of treatment in patients, 6 weeks of screening, and 6 weeks of post-treatment follow-up period. The primary endpoint in this study will be a combined assessment of function and survival called CAFS or CAFS at 52 weeks, so at this time point here, the double-blind, end of the double-blind period here, which is an estimation of survival and function. Function in this instance means the ALS-FRS score. So patients' survival and the decline of ALS-FRS score will be compared to the other patients in the individual groups, the Pegcetacoplan group, the placebo group, and they'll be scored, and the mean score between them, the two groups will be compared to determine the efficacy of the medication. So there are many secondary endpoints, including the severity and incidence of treatment-emergent adverse effects, the numbers who develop any significant depression at week 104, patients whose ALS-FRS score decline will be looked at from baseline to the 52 weeks and 104 weeks, change from baseline in percentage of slow vital capacity at week 52, and change from first assessment of percent SVC at home at week 52. Change from baseline in ALS assessment questionnaire at week 52, and then time to death, permanent tracheostomy or permanent assisted ventilation, which is the survival component of the CAFS. There are many other endpoints, including the European quality of life dimension, and then change from baseline in the Zarit burden interview, which is a caregiver interview to see how much burden they have in treating, in caring for people with ALS. Next slide. So the key inclusion criteria are the age at least 18 years, patients with sporadic ALS diagnosed as having definite or probable or laboratory supported probable ALS by the ALS scoring criteria. And these are important ones here now. Slow vital capacity of more than or equal to 60% are predicted at screening. And then onset of symptoms within 72 weeks or 18 months prior to screening. Total ALS-FRS score of more than or equal to 30 at screening. And then since Pegcetacoplan and similar compounds with complement inhibition activity can lead to an emergence of encapsulated organisms, there'll be a mandatory vaccination within 5 years against Streptococcus pneumoniae, against Neisseria meningitidis, and against Haemophilus influenzae type B. So all patients will have to either have had vaccinations against the 3 within 5 years, or they will be vaccinated within 7 days of starting their treatment. Key exclusion criteria include patients who are taking riluzole. They must be on stable dose for a month before they start the screening period, but they don't have to be on riluzole to participate in the study. And patients taking edaravone, which is the most recently approved anti-oxidative compound for ALS, they should be on stable dose for 60 days prior to the start of screening, but they don't have to be on edaravone. So, so I think that next slide. So yeah, so I think that you can go back to the first slide, the previous slide, please. Yeah, so I think that what we learned from this review is that complement is activated in patients with ALS. Complement activation in murine models of ALS happens very early before motor neuron degeneration occurs in the spinal cord and before axonal degeneration and loss happens in the ventral nerve roots. And the pathology then happens predominantly in the neuromuscular junction. There's deposition of C3 and C3 and breakdown products at the neuromuscular junction. There's also deposition of mammalian attack complex, or MAC, at the neuromuscular junction in the murine models of ALS. So modulating C3, the major central protein of the complement cascade, would be a relevant and a logical therapeutic target in patients with ALS. So I think that with that, I will stop and take any questions that there might be.
So thank you, Dr. Cannon. It looks like we don't have any questions in the portal right now. If anyone has any questions, you can type them in.
Yeah.
Okay. Looks like you have explained everything fantastically to me. So I really appreciate this. And again, thank you everyone for joining and listening to Dr. Tannen. We really appreciate it. And again, if you have any more requests for information, please visit the MERIDIAN trial website. We really appreciate it. Thank you very much.
Thank you so much, Scott. Thank you to the MDA.